everyone, welcome to my next video of gene transfer technique. So in my previous class we discussed gene transfer technique using agrobacterium. So as I already discussed in my previous class, agrobacterium, it's a biological mode of gene transfer method. So today we are going to discuss about the physical methods, what I have mentioned here, where we use for transferring of genes. Going on to physical methods, we have two important methods, or basically the techniques. One is electroporation, then the next one is gene gun method. So what is the advantage of physical method over biological method? The techniques what you people can see here, they are quite fast when compared to that of agrobacterium mode of gene transfer. And yes, electroporation as well as gene gun method, they have their own importance when it comes to matter of transferring the genes. So today we are going to discuss about the principles involved in the method, the factors which play an important role in the method, some of the instrumentation as well as the procedure. So let's go to the first topic of electroporation. Newman et al. In the year 1975 for the first time discovered electroporation method. Now what exactly is this electroporation? As the name itself indicates, we use electric current to create a pore within the plasma membrane. So in this technique what we are going to do is we are trying to use plant cell, basically the protoplast. Using this protoplast we are trying to introduce gene of our interest into the plant cell. So going on to its factors. Now as I told you, we will also be discussing about the factors. The factors are the time duration what we give the electricity. So that would be one of the important factor in successful completion of the transferring of gene. And along with that, we also check the kind of cell what we use. We cannot use any intact cell, a cell with cell wall. So one of the important factor is we do require protoplast. So taking a plant cell, removing the removing the cell wall, taking the protoplast, and then to this particular protoplast we go for electroporation method. So now what is the basic principle here? The principle lies in potential membrane or membrane potential. What we basically try to do here is we would be providing this protoplast with a particular span of electricity for a particular duration. What exactly happens? The plasma membrane present on this particular protoplast would undergo disintegration. If we keep on increasing the current, obviously the complete membrane would be degraded. So it is a key factor in order to maintain the proper time as well as the voltage, which play a major role in successful of this technique. Now going on to its instrumentation, I have not mentioned here, but yes, we do have something called as oscilloscope. Oscilloscope will basically try to check the pulse what we try to give. Along with that, we have a cell suspension chamber where we try to add cells as well as the gene of our interest. Along with that, we have a pulse generator. That means an electricity pulse which is generated for this particular technique. Moving on to the mechanism. If you try to see here, I have mentioned protoplast. So we will be taking a plant cell for which we will be doing protoplast isolation. Once we do this protoplast isolation, collect the protoplast. So that's one of the basic technique what we have to do for this electroporation method. Let me erase this off. Yeah. So once you have got the protoplast, my next technique is isolate the DNA. The DNA is of my interest. It may be a herbicide resistant, it may be a drought resistant, it may be a disease resistant. So the interest of my DNA would be accepted. What is the next protocol or what is the next procedure? Once we have got the protoplast as well as DNA, we will introduce them into cell suspension chamber. Now this cell suspension chamber would be having a electrode, positive electrode, negative electrode, and we have two important methods. This method basically includes low voltage, long duration, high voltage, short duration. Now as the name itself indicates, we basically go for a low voltage of 300 to 400 for a duration of say about 10 to 15 milliseconds. So the next what we have is high voltage short duration. Again here what we try to do, we give voltage of somewhere around 1000 to 2000 for a duration of 5 to 10 milliseconds. Either of LVLD or HVSD. My major aim is I wanted the protoplast which is intact should undergo disintegration. This integrated protoplast now will accept the gene of my interest which enters into the cytoplasm. As they enter into the cytoplasm, as we all know that there is pores in nucleus. Through this protoplast membrane they would enter into the nucleus. So once they enter into the nucleus, they will have a stable transformation. After this, we decrease the voltage which leads to reformation of the plasma membrane. So once the plasma membrane is reformed, we go for a stable integration, we go for cell wall generation, because this is an example of protoplast, not a complete cell wall, not a complete cell. So we go for cell wall generation. Once cell wall is generated, we go for callus culture. Once callus culture is done, we go for complete plantlet production, and the plant which is being produced is a transgenic plant which has a foreign gene which is of my interest. So that's with respect to your electroporation. Going on to the next topic, we have a technique called as gene gun method. Gene gun method is also called as biolistic method or particular bombardment method. Now why is it so specific, or what exactly is this gene gun? We try to introduce gene of our interest with the help of a microcarrier. This microcarrier would be shot in the form of a gun, in the form of a bullet, to the material what we wanted to transfer the gene. Now let's enter into this topic. Gene gun method was introduced in the year 1983. So what exactly is this, or what exactly happens with this particular technique is, it can transfer the gene into subcellular organelle. Now what exactly is subcellular organelle? Something like mitochondria and chloroplast. If I wanted a gene to be introduced into chloroplast or mitochondria, I can use this particular method. I don't want any kind of special protoplasts like this. So a normal cell, a cell with its own cell wall, can also be used for transferring of gene. Now going on to its instrumentation, we have a helium gas chamber where we try to allow the gas to flow. We have a rupture disc, we have a macrocarrier, we have a microcarrier, we have a stopping screen, we have the explant material. Now if you try to check here, the distance between stopping screen as well as explant, it should not be more than 10 angstroms. So that should be the distance between the stopper screen as well as the explant. Now what we basically try to do, we try to take either gold or tungsten of 1 to 3 micrometer diameter. Now this gold or tungsten of 1 to 3 micrometer in its diameter, they are called as microcarrier. So now what exactly they do? To this gold or tungsten, we incorporate the gene of our interest. That is, DNA would be added onto either gold or tungsten. Now why specifically gold or tungsten? Both are non-reactive. They should not react, or they should not show any kind of chemical properties, or they should not show any of the process to combine with the DNA and followed by degradation of this DNA. So that's the reason we go for an inert molecule, either gold or tungsten, and the diameter should not be more than 1 to 3 micrometer. Now attachment of DNA to this gold or tungsten, we use spermidine. Spermidine is a special chemical which is used to coat this microcarrier with the DNA. Now let us go into the actual process. So I have helium gas here. Initially what we try to do, we try to pour or we try to send helium gas onto this particular chamber. So once helium gas is being introduced into gas chamber, as the pressure keeps on developing, there is rupture in the disc or the disc breaks, and hence it is called as a rupture disc. So as the disc ruptures, something like this, all the pressurized helium gas will now strike to macrocarrier. So once they strike to macrocarrier, this macrocarrier is a movable platform. So now what is the importance of this movement? So let me erase this. Okay. So let me also erase this. So once they fall onto this macrocarrier, this macrocarrier will start moving in this particular direction. As they try to move downwards, we have something like a stop screen, or it's also called as a stopper screen. So with a high velocity, macrocarrier would come down, and because of the presence of the stopper screen, they stop there. But the actual process begins now. As they come down and you have this stopper screen, the molecule which has the DNA, that is the microcarrier, will fall onto the material. And the material what we try to keep here is an X-plant, basically plant cells, or in the form of a callus, which accepts the foreign DNA. So in this way, the DNA enters into the plant cell, and followed by this plant cells, if we try to grow them, we get a stable kind of transformed plant material, and if we develop that particular plant, we are going to get a transgenic plant. So either gene gun method or electrofusion method have their own applications. So thank you for watching. Have a nice day.